Hey, microbiologists. Welcome back to class. We are in chapter 2. We have just finished discussing carbohydrates. And the next biomolecule we're going to talk about are lipids or fats. Nice, yummy butter and all of that good stuff. So in carbohydrates, we saw we had a carbon hydrogen oxygen ratio of 1 to 2 to 1. In lipids, we have a lot less oxygen, so we're not even going to count the oxygen there, but the carbon to hydrogen ratio is 1 to 2. So if I tell you you have a lipid that has 12 carbons, you could tell me it has 24 hydrogens in it. The defining feature of lipids, the big unifying trait they have, is they are non-polar. So all of these carbon-hydrogen bonds are going to share their electrons equally. You don't have any electrons hanging out at one side more than the other. So they are not going to form hydrogen bonds with water. They are not going to interact with water at all. They're going to be insoluble. Or in other words, they are hydrophobic. This would include fats, oils, and waxes. So if you've ever tried to mix vinegar and olive oil to make a salad dressing, you notice they don't mix too well. They separate from each other. Or if you've been in a parking lot after it rains, you can see if there's a car that's been leaking some motor oil, it'll sit on top of the puddle. It doesn't mix in because our lipids are hydrophobic. They just don't mix in water. That also means that lipid molecules are not going to mix in with the water of your blood. So to be transported through your body, they're going to need some specific transporter that will somehow combine with that lipid to get it through your blood system to your body. Lipids include a bunch of these nice little chemical sounding names. The ones we're going to talk about most is phospholipids. Those are super important in our cell, and we will talk about phospholipids for the rest of the semester. We'll also talk about fatty acids. They're kind of the basic model of lipids. Steroids are important. We'll talk about it a little bit. We will probably not really talk about these other three much. I'll mention them here or there, but really phospholipids, fatty acids, and steroids is where I would put my time studying about lipids. One thing that lipids do is they are the primary components of cell membranes. These phospholipids here make our cell membranes. And the reason they're so good at being cell membranes is because they are hydrophobic. That hydrophobic lipid is not going to dissolve into the water inside the cell. It's not going to dissolve into the water around your cells. It's going to maintain its integrity as a lipid to create this cell membrane. So lipids, because they're hydrophobic, they make great borders for our cells. Simple lipids would be things like fats or triglycerides. They're called triglycerides because they have a glycerol with 3 fatty acids on it. They're formed by dehydration synthesis, broken by hydrolysis, change of hydrocarbons. They can be saturated or unsaturated. I feel like I don't really want to read the slide to you, but I would rather just talk about it on a picture. So here's the information. You can get to the PowerPoint if you want. Here's what a lipid looks like. So here is a glycerol. Here are 3 fatty acids. This dehydration synthesis, you can have a dehydration synthesis here, dehydration synthesis here, dehydration synthesis here. So you've 3 reactions, add these 3 fatty acids, 3 waters run off to do whatever they need to do. Or if you want to break it down, you can do hydrolysis to break each one off. End up with this water molecule incorporated here in these bonds. Then another thing that I talked about here is let's talk about hydrocarbons before we do that first. So change of hydrocarbons, that means it's carbon and hydrogen, carbon and hydrogen, carbon and hydrogen. That's what this whole tail is. Carbon and hydrogen, carbon and hydrogen. So they're hydrocarbons. Okay, and then it also talked about saturated and unsaturated. So here you can see each carbon has 2 hydrogens, 2 hydrogens, 2 hydrogens. That's being saturated. Every time you have 2 hydrogens, it's saturated. If you come over here, you see this carbon has 1 hydrogen, this carbon has 1 hydrogen. That's unsaturated. They both just got rid of a hydrogen and made a double bond with themselves. So instead of bonding to each to hydrogen, they're bonding to each other. That's unsaturated. Whenever you have a double bond, your fatty acid is unsaturated. And one weird thing it does is this double bond gives it this little bend. See how it suddenly bends here? That bending is happening because of that double bond. So saturated fatty acids are going to lie nice and flat against each other, so they're able to pack solid. These are going to be things like butter, things that are solid. Unsaturated fatty acids, they get that kink, so they don't pack so solid. They're going to be a little more liquid. Things like olive oil. Unsaturated fatty acids tend to be more plant-based or fish-based, while saturated tend to be more red meat-based. Lipids can be more complex, so when you go from having just carbon and hydrogen to involving oxygen and phosphorus, nitrogen, and sulfur. One complex lipid would be phospholipids that make up our cell membranes. Phospholipids are kind of cool because while their lipid part is hydrophobic or water-fearing, it doesn't interact with water. All these little tails here are hydrophobic tails. They also have this head, this phosphate head, that is hydrophilic. And so that phosphate head that's hydrophilic is willing to interact with water. So while all of these fatty acids are making a nice boundary so that your cell doesn't dissolve, so that you can define what is in and out, this phosphate head, because it's hydrophilic, it's willing to interact with the water inside the cell. So you don't have some weird refusal to interact. Your phosphate head will interact with water inside the cell. These phosphate heads here will interact with water outside of the cell. So it's a very nice boundary for our cells because it is both hydrophilic and hydrophobic. Another type of lipid is a steroid. Steroids are these 4 rings, so A, B, C, D, these 4 rings. They have a hydroxyl group attached to one ring, and then they'll have different functional groups attached to them, so all of this stuff can vary. Steroids are incorporated into our cell membranes, and there they help keep our cell membranes fluid. They keep our phospholipids from packing together too tightly and freezing in place. Instead, they keep a little bit of space so your membranes kind of have space to be moving around and stay fluid. Steroids are also the basis of a lot of other chemicals that go on in multicellular organisms, although that's less relevant to our discussion of microbes. So at this point, you can tell me how simple lipids differ from complex lipids. You could tell me what are the properties of a lipid. You could describe to me phospholipids, what their structure is, why they make great cell membranes. You can discuss to me what the structure of a steroid is. Okay, I'll meet you back for our next mini lecture about proteins. See you soon.